Here is a very quick video because it's not a very difficult enzyme compared to the restriction enzymes. This is our second out of 3 enzymes that we talk about. This one is called ligase, or sometimes DNA ligase. You can call it either one. So what is ligase? What is the point? Lig or lig, that little prefix there, that means to bind. Ase is going to be enzyme. So when you cut enzymes using restriction enzymes or restriction endonucleases, you want to be able to stick them back together. But just putting them back together and relying on them to attract to each other won't really work. So what we need is we need another enzyme that is going to stick the pieces of DNA together. Now, if they're sticky ends, it's only going to join complementary ones. Yeah? If you've got sticky ends like this, you can only, so with sticky ends, you can only join complementary sequences, DNA sequences. Yeah, but if you had blunt ends, you could join any 2 blunt ends together. So what is ligase? Ligase is simply the enzyme that is going to rejoin your backbone, so that it works on joining together the phosphates and the sugars in the backbone of that DNA. And therefore you end up with a nice straight piece of normal DNA and you wouldn't know any different. From this, you can now go through normal DNA replication. You can do a whole bunch of other processes. But ligase's job is to join 2 pieces of DNA. That's it. Join 2 pieces of DNA. You can see here, this is just an example that we're about to kind of unpack in detail. You can see that EcoR1 was used to cut out this pink piece of DNA. They also cut a circular piece of DNA here. Now, when you put them back together, yeah, you can see that they have complementary sequences because they use the same restriction enzyme. And as a result, they can join them back together. So they have to be, I'm just going to get rid of this mismatch stuff because you don't need that. These are joined with DNA ligase. If you leave out the DNA, that's fine. You can just call it ligase as well. And that's all I have to say about ligase.